So it's another Sunday and another month, and we are back at the KDK talk. And I take this opportunity to welcome all of you who have joined us online on behalf of Jawaharlal Nehru Planetarium, Bengaluru, and ICTS. So those were the days when we used to have hundreds of people gathering at the planetarium on a Sunday evening for a similar program and have a wonderful live interaction. It's been more than 1.5 years, and it looks like it has been a long time ago. But then the things are looking up, and we hope that at the same rate at which the pandemic is subsiding, we may be able to have the Kaapi with Kuriosity talks at the planetarium in person very shortly. And meanwhile, let us enjoy at least the online facility that we have to get the scientific ideas across. And we welcome you all and thank you for the support that you have extended during all these months. And we have a wonderful talk titled Autism and Astrology. And Astro in quotes is something very interesting. And I think Professor Sumantra Chattarji is going to, during the course of the talk, unquote Astro part of astrology. And I now request Professor Vijay Kumar of ICTS to introduce the speaker. And we look forward to a wonderful talk by Professor Sumantra shortly. So, Professor Vijay Kumar, over to you.
Thank you, sir. It's a great pleasure to introduce Professor Sumantra Chattarji, or Shona as is popularly called. So Shona did a master's degree in physics from IIT Kanpur. He then switched to neuroscience, and he got a PhD from Johns Hopkins and the Salk Institute. And he spent a couple of years doing postdoctoral research at Yale and MIT, and then he joined NCBS, the National Center for Biological Sciences, in 1999, where he's a senior professor now. Using animal models, Shona's lab has explored the neural basis of many emotional problems related to autism and stress-related psychiatric disorders. His research has shown that prolonged stress leaves its mark by enhancing both the physiological and structural basis of synaptic connectivity in the amygdala, and thereby triggering the emotional symptoms observed in stress-related psychiatric disorders. Shona's lab also studies synaptic defects and their reversal in Fragile X syndrome, which is the leading identified cause of autism. Shona has been well recognized for his outstanding research work. He was awarded the Global Champion Award by the Fragile X Research Corporation, Research Foundation USA. He's also a fellow of the Indian Academy of Sciences and the International Union of Physiological Sciences. And last year, he was elected a lifetime member of EMBO in recognition of his remarkable achievements in the life sciences. And so today he's going to tell us about new insights on autism and astro, again within quotes, astrology, new insights from recordings in the human brain cells. Before I hand over, please feel free to put in your questions in the Zoom chat and also on the YouTube chat. People who are watching it on YouTube, our team will collate the questions and will answer them towards the end of the talk. So with that, it's again a warm heart feeling to welcome Shona. And Shona, the Zoom screen is yours.
Thank you very much, Vijay, and thank you to all of the organizers who have put in the hard work to put this together. And I've always been a big fan of this forum and what it achieves in terms of outreach and scientific communication. So it's a genuine honor to be part of it this time and talk to you about some of our recent work, all of which is right now unpublished work that I'll share with you. I'll start off by talking about a major challenge that we face in the health sciences, which is that studying the healthy brain itself in human beings is quite challenging because of the enormous complexity of the human brain with its billions of neurons and their connections and the many functions that it performs. And we, unlike other organs of the brain, we do not always have direct access to living tissue from the brain, meaning we cannot take biopsies like we can do from other organs, for example. So this poses some significant technical barriers in terms of trying to see what's gone wrong inside the brain with non-invasive techniques. There are, of course, some ways of looking at the brain, as you well know. One can look at post-mortem brain tissue and compare healthy brains with diseased brains. But this is more like archaeology of the brain. The damage has already been done, the patient is diseased, and then we look at what may have happened in the past. That's not very helpful. You can also look at the living brain by doing structural magnetic resonance imaging or MRI, which is often done to diagnose disorders of the brain. And as well as we can do functional MRI, which gives you insights into aberrant patterns of activity in the living brain as it performs certain functions. But unfortunately, all of these techniques have pretty serious limitations because their spatial temporal resolution is rather limited. You can look at best a few millimeters of spatial resolution. And so the bluntness of the tools is a major barrier in trying to go deep into understanding what the problems are in the brain. And we need to have much higher resolution analysis of cells and molecules to really understand what may have gone wrong, because that's the level at which drugs act. And so if you're to design new therapeutic strategies, we need to understand what's going wrong at the level of cells and molecules, which is not possible to do right now in a living human brain. So we make compromises. We try to find surrogates for the human brain. And what we do is we switch to animal models of rats and mice and try to create disease models in these animals, because that allows us to analyze the problem across biological scales, all the way from behavior down to the level of molecules and genes. And so we now have the possibility of doing high resolution analysis of cells and molecules in such disease models in animals. You can do sophisticated manipulations to see if certain interventions work. You can do all of it in living tissue and cells, which is great, but it ain't human. It's a rat or mouse. So there's a trade-off that we have to live with. And this trade-off is what the current state of affairs has been till a few years ago, which is that we create a disease model in an animal and then treat that disease in the animal, find promising drug candidates in the animal, and then take it back into human trials and hope that the same drugs now work in the human disease and gives us some benefits. And that's where, unfortunately, much of this whole paradigm breaks down because what worked in rats or mice quite often in case of brain disorders does not work well enough in humans. And so what we find after a very prolonged and expensive exercise is that humans are bad models for rodents. And so this becomes a very costly affair and time-consuming affair, as shown in this slide. The whole pipeline of drug discovery, where you start with thousands of compounds on the left, and then you narrow it down with preclinical animal model studies like the one I showed you, and narrow it down to a few hundred compounds. Then you go into clinical trial after safety and everything else is taken care of. And then eventually maybe one or two compounds, if you're lucky, may be approved by the FDA, which means it takes a very long time, 7 to 10 years often. And then there's a high rate of failure and it becomes very, very expensive. So most drug companies, major drug companies, shy away from this business now. And compared to, say, cancer and other disorders, there have been very few success stories in terms of new drugs for brain disorders. So this is the current state of affairs, a rather sad state of affairs. But there's now a heightened need to try to combine sort of best of both worlds. If we could have human cells, but cells and not, post-mortem tissue, and they're still from human beings without killing a human being, that would be the best of both worlds. So is that possible? And the answer is yes, and largely because of the pioneering work done by Yamanaka and others who have pioneered the technology of generating human stem cell models over the last more than a decade now. The Nobel Prize-winning work by him and others is now allows us to actually have human brain cells from healthy individuals as well as diseased individuals, grow them in a dish. And now when you're studying the aberrations in a brain cell, you're not studying that in a mouse or a rat brain cell, you're doing it in a human brain cell. And that's what I'm going to talk about today. And I'll pick a specific example for this, which is the example of Fragile X syndrome, or FXS in short, which is the leading genetic cause of intellectual disability and autism spectrum disorder. And it's something that's been studied extensively using animal models, leading to multiple drug trials, all of which, unfortunately, did not give promising results at the end of the trials. Now, Fragile X syndrome is caused by the absence of a specific protein called the Fragile X mental retardation protein, or FMRP for short, which is produced by the Fragile X mental retardation gene. And a mutation in this gene is what causes the shortage of Fragile X mental retardation protein, which then gives rise to the disorder. It's more prevalent in boys than girls and has a range of debilitating symptoms, such as developmental delay. Some percentage, about one in 4 individuals will have some form of seizures, high social anxiety, unstable mood, very hypersensitive in terms of sensory inputs, below average IQ, and attention deficit is another hallmark of these disorders. So it's pretty difficult set of symptoms to manage for these kids. So the work I'm going to talk about is done by 3 amazing young scientists. Dr. Shreya Sharma, who recently got her PhD from my lab, Dr. Rakhi Pal, who is a fantastic expert in stem cell biology, and she knew a lot more than any of us when she joined the group. And she's really driven many aspects of this project herself. And she's been ably accompanied by Bharat Reddy, who's really taking care of another crucial aspect of the study and has done a phenomenal job. And all 3 have worked through the pandemic to give rise to a bunch of data and results that I'll talk to you about today. So thanks to them for making this possible. So what they have done is Rakhi and Bharat have, with help from our collaborators, have devised a protocol for generating cortical neurons from what is called human pluripotent stem cells. This is the technology developed by Yamanaka and colleagues, where you can basically take skin cells, for example, fibroblast from an individual, living individual, and then go through some amazing molecular cookery where you add certain factors, the Yamanaka factors, to get pluripotent stem cells. The word pluripotent tells you what they mean. These are now cells. They have the potential to become any cell you want in the body based on how you treat them with certain chemicals and reagents. So through a whole bunch of steps that I will not go through, eventually you arrive at what are called cortical neurospheres and then neural precursor cells, and then they differentiate into human cortical neurons. This is a process that takes months to do in a dish, but and pretty demanding protocol. But when it works, it's absolutely fantastic because you essentially have the individual still intact. We haven't touched him or her in any way, except for those little skin cells that were given by the donor can now be put through this pipeline such that at the end of it, you actually have human cortical neurons originating from that individual, meaning they're the same genetic makeup as a living individual without ever diving into their brain. And so, and there's another way now that you know, a couple of years ago, there was a Nobel Prize given for gene editing technology, CRISPR-Cas9. We also have ways of deleting, sort of creating the same mutations that give rise to Fragile X syndrome by acting on the Fragile X mental retardation gene, FMR1 gene, and then create gene-edited knockout cell lines as well. So both from patients as well as from through gene editing technology, we can create Fragile X neurons, if you will. And for the sake of simplicity in this talk, I will interchangeably refer to disease, sort of patient-derived cells, as well as these gene-edited cells as Fragile X neurons. Okay? But the origins of this are shown in this slide. And so we have multiple healthy lines, which are from normal individuals who do not have Fragile X, and we'll refer to them in this kind of work as control neurons. And we also have Fragile X neurons, either gene-edited or from patient lines. So the first set of experiments I'm going to talk about have to do with growing these neurons in a dish, okay? And so it's called a culture of neurons. and you grow them. You take the human pluripotent stem cell derived human cortical neurons and grow them alongside another class of brain cells, which are less talked about, and they're called astrocytes. So astrocytes have long been viewed as playing a supporting cast in the brain. They are supporting cells that provide healthy, support healthy function and communication between neurons, and have largely been overlooked in the field till more recently. So this is now a culture of brain cells that has two populations. It has the human cortical neurons alongside astrocytes taken from the mouse brain. So we have mouse astrocytes co-cultured with human cortical neurons, and they're all grown in a dish. And this is the population we're going to first study. But when I say study, we record the electrical signals from the neurons in this culture. So we do not record from the mouse astrocytes, we only record from the human cortical neurons in the company of the mouse astrocytes. And when I mean recording, I'll tell you exactly what we mean in terms of the electrical signaling. So I'll divert a little bit and give you a basic sort of primer in terms of neuronal communication, how neurons communicate. Here you see two neurons, their cell bodies shown here. Now neurons have a cell body, which is the center of its information processing, and it has two classes of branches, just like a tree. One branch is called the axon, which is the output end of the neuron, meaning it sends out electrical signals to other neurons through that branch. Now, when this signal is sent out, it is then received by other neurons, and the receiving neuron receives these signals on the second class of branches, which are called dendrites, which is the input end of the neuron. So what happens is the following. If this neuron gets sufficiently excited, it will fire a particular kind of electrical signal called an action potential, which is nothing but a sudden spike, a surge of voltage in the neuron. A neuron normally rests at a very negative potential compared to the outside world. So it is sitting at minus 70 or minus 80, around minus 65, minus 70 millivolts. And when it gets sufficiently excited, that voltage changes suddenly and surges upwards and spikes towards the positive side and then comes back down quickly. And this spike is called an action potential. And that's the output signal of the neuron, which travels down this axon and arrives at the end of the axon terminal. The axon ends at some point. At the end point of these axon branches, it makes contact with the dendrites of the next neuron. Now, the dendrites are the receiving end of the next neuron, and that's where the signal is transferred from the first neuron to the second neuron. And the transfer happens at a contact point between the two neurons, which is called the synapse. So this is where information transfer happens. That action potential, the electrical signal, arrives at the terminal of the first neuron, releases packets of chemicals called neurotransmitters. Those transmitters now diffuse across the gap and then goes and binds to specialized protein molecules in the next neuron and opens up, triggers a signal in those neurons, which are the synaptic signals that that neuron receives. And those synaptic signals then get transferred along back down to the cell body of the next neuron. If all of those summate together to cause enough excitation in the next neuron, it will again fire an action potential. So this is how electrical communication happens from one neuron to another. So what does an action potential look like in real life? This is how it looks like. So you can see that the units we're going to talk about for today is in millivolts and in a time scale of 1 to 2 milliseconds. So this is about a 2 millisecond spike or surge in membrane potential. These individual action potentials, as shown here, or for short, we call them spikes. These spikes are the essential currency of communication between neurons in the brain. So this is how billions and billions of spikes are propagated from between neurons to communicate signals between neurons. And that's where the information is contained. And for today's talk, I'll mostly only show you these spikes. So I'm going to keep the biology very simple. You'll only see signals which are electrical signals, either changes in voltage or changes in current. And I guarantee that you will see no other biology other than this. Okay, so I'm hoping that changes in voltages and currents should be sufficiently simple for the string theorist in the audience to understand. So spikes, what happens? So if you now record from one of these healthy human cortical neurons in a dish using an electrode that is positioned on the cell body of these neurons and see how it fires spontaneously, here's what it looks like. Here's a healthy cortical neuron which fires. You can see these vertical lines. Each of these vertical lines is a single action potential. So what you see is that the cell is quiet for a while, and then when it gets sufficiently excited, it fires not one action potential, but a tightly bundled burst of action potentials, as shown here. Then it goes quiet, and it stays quiet for a long time, and then again there's a burst of action potentials, so spikes fired by this neuron. So this is the pattern of firing in a healthy cortical neuron. Compare that. Yeah.
There's a quick question from Arush Murali.
Okay.
And he refers to the slide that you were showing earlier.
Mm-hmm.
The question is, before the spike in potential, there seems to be a smaller mini spike. Is this a measurement error or a feature of the neurons?
Oh no, this is just I think a little blimp of it. It's not the action potential, but it may be a bunch of synaptic events that came and coalesced together. Or it could be a reflection of a stimulation and it's a stimulus artifact. It could be one of the two. Okay.
Thank you. Yeah, please continue.
Yeah. And so what you'll be seeing is this is a single action potential. If you squeeze it in time, you will have and record for seconds. Of course, individual action potentials will look more like a line, and that's what you're seeing in the next slide. So a normal, healthy cortical neuron fires these prolonged bursts of action potential separated in time. Compare that and contrast that to a neuron from a Fragile X patient, which fires many such bursts, abnormally high number of bursts, but the individual bursts you'll notice are rather short. They're nowhere near as prolonged as these, the healthy neurons. So what you see is very, very clear in terms of very distinct firing patterns. The healthy neurons fire few prolonged bursts with gaps in between, and the Fragile X neurons fire too many bursts, but they're too short. So the aberration is very clear. So you can actually expand one of these bursts, and you can see there's hundreds of these action potentials being fired by a healthy neuron. Compared to this, you can look at the Fragile X neuron. It's not doing too well. It's barely able to sustain a few action potentials, then it dies out, but it's firing a whole bunch of these bursts. Okay. So you can take a gene-edited line where you deliberately do CRISPR-Cas gene editing to create a Fragile X neuron. You see the same distinct patterns of firing, or you can take it from a patient and you see the same thing. So this gives you confidence that this is indeed a genuine aberration in firing patterns. So if you do these recordings, what do we measure from them? We have to quantify it. So we do two simple things. We look at the burst duration in terms of the time spent in such a bursting firing and the burst number. How many numbers of bursts does it fire in a 10-minute window? So it's very simple. How long are the bursts and how many are fired within 10 minutes? Okay. So if you do that, here's the burst number per 10 minutes plotted on this graph. And you can see that the red bars, which are indicative of the aberrant firing by the Fragile X neuron, there are too many bursts. Whichever way you do it, gene-edited, patient-derived, they're firing too many bursts. On the right-hand side, we are plotting the burst duration in seconds. And you can see that in all of these cases, the red bars are much below the black bars, meaning they're firing much shorter duration bursts compared to the healthy neurons. So this is the basic measurement, basic finding is that the Fragile X neurons are firing more frequent bursts, but of shorter duration. So what's going on in these human neurons, Fragile X neurons, is the question, what's gone wrong? What's the underlying reason for this? Now, there, of course, in biology, there are always multiple factors. There are many things that can cause the same thing. So we wanted to go with something that we could start with and get some traction on this question. So we went back to the literature and found one particular study, along with a few others, which suggested that such changes in excitability could be caused by what is known as a particular type of sodium current. Now, as I told you, these currents are generated by ion channels, which are basically specialized protein molecules, which are like gates embedded in the membrane of the neuron. And these gates are normally shut till they're excited or stimulated or activated. And then the gates open up and the door opens, and now it allows ions to flow through. And in this case, it allows sodium ions to flow through. Sodium is a positively charged ion. And if you have movement of charged particles, you have current. And so what you see, you can record as these channels open up and the ions move through, current flows through, and you can record these currents from the neuron using electrophysiological recording techniques. So we focused on this particular persistent sodium current because it has been shown to be changed in a mouse model of Fragile X in the past. So we thought, well, this is a reasonable guess to start with. So Shreya, who's been doing all of these recordings, sort of basically adopted a protocol from previous literature, which involves, the cell is sitting at minus 80 millivolts at resting. You push it down to minus 100 millivolts and then slowly ramp it up at a rate of 20 millivolts per second, all the way up to minus 20 millivolts and then back up. Essentially, you're applying a voltage ramp on this cell and forcing it to follow it, as a result of which you get a certain pattern of activity in terms of the current. So you inject a depolarizing ramp and then record. Then you add a sodium channel, a generic sodium channel blocker called TTX, wait for 5 minutes. Then you inject another depolarizing ramp and record. And then you subtract the two traces that you have recorded and you get what is called the persistent sodium current, or INap for short. INap standing for the I standing for the current, Na standing for the sodium, P is for persistent. So you can now isolate and selectively record the persistent sodium current from healthy control neurons, and it looks something like this. So this is the time axis in 400 milliseconds, and on the y-axis you have the current is measured in as small as hundreds of picoamperes being 10 to the power minus 12 amperes. Okay, so this is how the healthy neurons persistent sodium current looks like. We measured this amplitude right here, as shown here, the peak value. And compare that to the Fragile X neuron, it's missing. It's just not there. It just doesn't get to the same level. So there's this very significant reduction in the persistent sodium current in the Fragile X neurons. So you can ignore the left-hand side, but just remember that basically the persistent sodium current suffers a significant reduction in the Fragile X neurons. Okay, and I want you to sort of remember this because we'll come back to this at a later stage. So if you now remember this summary, in control healthy neurons, they fire prolonged bursts, but few of them. Fragile X neurons fire too many bursts, but they're abnormally short. And controlled healthy neurons have normal persistent sodium current, whereas the Fragile X neurons have a significant reduction in the same persistent sodium current. So this is the state of affairs as of now, what I've told you. So far we have used human neurons alongside mouse astrocytes. Next, the question was, what happens in human neurons that are co-cultured with human astrocytes? So now we are creating an all-human culture system where you have human cortical neurons commingled, mixed up with, grown together with human astrocytes. Okay? And so it's the same programming, same protocols again. We now have a protocol where Bharat can generate human astrocytes as well, alongside Raki's protocol of human neurons. And then we co-culture them. And now for some astrology and demystifying my title. So now we have human cortical astrocytes co-cultured with human cortical neurons together, kept together, and then 8 weeks later, we are going and doing the same wholesale patch clamp recordings using electrodes to record from the neurons, not the astrocytes, only the neurons, and see what happens. So earlier we had this finding from human neurons with mouse astrocytes. Now we have human neurons with human astrocytes. And now you realize there are 4 different possibilities, right? You can have Fragile X neurons with Fragile X astrocytes or control astrocytes, and then control neurons with Fragile X astrocytes or control astrocytes. So 4 different combinations, right? So if you have control healthy neurons co-cultured with healthy astrocytes, you have the same old firing, the healthy firing pattern I showed you earlier. Prolonged bursts, but few of them. Here's a control healthy neuron, genetically a healthy neuron, but it's firing like a Fragile X neuron, meaning too many bursts, too short. Why is that? You know why? Because it's been co-cultured with Fragile X astrocytes. Bad company. And now it's firing like a Fragile X neuron, although genetically it's actually a healthy neuron, but just because it's in the vicinity of Fragile X astrocytes, it's now firing like a Fragile X neuron. If you take a Fragile X neuron along with a Fragile X astrocyte, they fire like Fragile X neurons with aberrant firing pattern. And quite remarkably, if you take a Fragile X neuron co-cultured with a healthy astrocyte, that Fragile X neuron now shows normal firing. This, you realize, is of great interest from a therapeutic drug discovery point of view, because this is actually a diseased neuron, which is firing like a healthy neuron only because it's in the company of healthy astrocytes. So the healthy astrocytes is able to override the diseased firing pattern in the Fragile X neuron, which is quite remarkable. So basically, you'll see what the pattern is emerging is that control neurons control astrocytes, the black firing pattern. Fragile X neurons and the Fragile X astrocytes, the red firing pattern, the aberrant firing pattern. But Fragile X neurons with control astrocytes fires like a normal neuron. And a control healthy neuron, despite being a healthy neuron itself, fires like a Fragile X neuron just because it's grown up with Fragile X astrocytes. This is the quantification. You can see that the burst number in control neurons is low, right? Remember, control neurons with control astrocytes is the normal firing pattern. Burst numbers are low. But the same control neuron, when grown with Fragile X astrocytes, it starts firing too many bursts. And the burst duration, vice versa, it's prolonged in control neurons co-cultured with control astrocytes. But the same control neuron, when put in touch with Fragile X astrocytes, the burst duration goes down. And of course, you have both together, then it stays the same. And conversely, you can take a Fragile X neuron, and just by growing it with a control astrocyte, you can reduce the burst number, and you can increase the burst duration. So this is quite astonishing, that the astrocytes basically determine what goes on in the neurons. So next question is something we tried was quite crazy. We said, do you really need to have the astrocytes in the dish? What if the astrocytes are not even present in that network, right? So take the astrocytes away from the equation and just take the soup from it and see if that's enough. So you don't physically have the astrocytes in the network along with the neurons. So this is what I call, in southern India, this is a very easy talk to give, because this is what I call the rasam experiment. So these astrocytes are grown in a conditioned medium with a lot of stuff in it. It's like a soup, and it's called conditioned medium. And what we do is we grow the astrocytes, either control or Fragile X, by themselves, and then take that soup from that culture and then pour it into the neurons, okay? So instead of the astrocytes themselves, we just take the soup. So take the astrocytic conditioned medium and put it on the dish in where the neurons are growing, and then you go and record from the neurons. These are now neurons which are by themselves, but they have grown up in either the healthy or the Fragile X astrocytic rasam. That's the experiment. So now this is something quite remarkable. Here you have a black neuron, meaning a healthy neuron. Genetically, it's a normal neuron, but in the presence of the astrocytic conditioned medium taken from a Fragile X astrocyte. See the white here symbolizes the fact that the astrocyte is missing. It's not there. It's only its soup that's there. And that is enough to make a normal, healthy neuron to fire like a Fragile X neuron. That's what's shown here. It's now genetically, it's a normal neuron, but it's fired like a Fragile X neuron just because it's been exposed to the Fragile X astrocytic rasam. Conversely, you can take a Fragile X neuron and just pour on the healthy astrocytic rasam, and it starts firing like a normal neuron. So this is quite remarkable. So it's the astrocytic condition medium alone can determine how the neuron will fire, right? You don't actually need the astrocytes anymore. So the astrocytic rasam is enough. So that's the major breakthrough, because this means that that rasam has something in it as secreted by the astrocytes that has the magic potion. That's enough to convert the firing both ways. It can make a healthy neuron fire like a diseased neuron, and more importantly, a diseased neuron can be made to fire like a healthy neuron. So this is what's summarized here. Rasam is good in life as well as in science. If the control neuron with a control astrocytic rasam is fine, fire like normal. Fragile X, fine, fires like a diseased neuron. But the stunning finding is that a healthy neuron with Fragile X rasam fires like a Fragile X neuron, and vice versa. And you will see the firing pattern's color tracks the color of the rasam. This is genetically a Fragile X neuron, but it fires like this because it matches this, and it matches this. So it's the rasam's character that decides how the neuron will fire. So that obvious question next is, what's in the astrocytic rasam, right? And now that's a complicated question, because these media have lots of different ingredients, lots of stuff goes into it. And so it's what I call a needle in a haystack kind of problem. Where do you start, right? And there are many possibilities. So again, we go back to the drawing board and look at past research and the literature from other people's work. And to make a long story short, we got some promising clues from previous work by others. One of them involved the finding that in children with autism, the plasma concentration of a particular molecule called S100 was high. Now, this is a molecule that is selectively found only in astrocytes and is secreted by astrocytes. And so this same S100 was shown to alter firing, action potential firing patterns in neurons in a mouse model, okay, in a mouse study. So that was a second line of evidence that gave us hope. And then in a Fragile X mouse model in the cerebellum, people found that the same astrocytic molecule, S100 is actually abnormally elevated. So we thought this is a good guess to start with. So we had a working hypothesis, where we said that, okay, the Fragile X disease condition, the Fragile X firing pattern, will be equivalent or representative of high levels of S100 If you have high levels of S100 that's the Fragile X condition. You have low levels of S100 you have the healthy condition. And we picked these numbers fairly arbitrarily from previous literature. We assigned that the high level means 1 millimolar concentration of S100 and the low levels was equivalent to a much lower concentration of 100 micromolar of S100 And then Shreya now did a different set of experiments where, while she's recording from a neuron, she collects a baseline firing pattern. Then she adds the 1 millimolar, the high concentration of S100 in that same bath in that recording in the medium for 20 minutes. And then again, after a while, continues to record from the same neuron and see if and how that neuronal firing pattern changes after the addition of the S100 in the high concentration, the 1 millimolar. So within the same cell, before and after addition of 1 millimolar high concentration of S100 That's the experiment. The recordings are just the same. So here's now a healthy neuron with a healthy astrocyte firing the healthy pattern of firing, showing the healthy pattern of firing. And once you add this high concentration of S100 it's the same exact neuron, but it's now firing like a Fragile X neuron. And that's quantified here, shown here which is very convincing, and it's quantified here. The burst duration is to be high in the healthy neuron. It becomes much shorter after the addition of 1 millimolar S100 beta. And conversely, the low burst number seen in control neurons gets higher after the addition of the same S100 beta. So the firing flips just by adding 1 millimolar of S100 beta. Now, in Fragile X neurons with Fragile X astrocytes already in the disease state, so adding another 1 millimolar of S100 beta makes no difference. It's already at the disease state, so the firing remains the same as shown on the right-hand side. If you take a control neuron with Fragile X astrocyte, we already know that the astrocyte determines the neuronal firing. So although genotype-wise, this is a healthy neuron, because it's in the company of the Fragile X astrocyte, as expected, as I showed you earlier, it's firing aberrantly. And it's already in an aberrant state, so adding S100 beta at 1 millimolar concentration makes no difference. It stays the same. It's already in the disease state. However, if you take a Fragile X neuron in the company of a healthy astrocyte, it fires like a healthy neuron, as shown here. And now, on top of this healthy firing pattern, if you add 1 millimolar S100 beta, it starts firing like a Fragile X neuron. So again, converts. So the findings from these studies tells us that high, meaning 1 millimolar of S100 beta, a specific molecule found in astrocytes and secreted by astrocytes, is able to switch normal firing into aberrant firing patterns in these neurons. So essentially, the high levels of S100 beta does represent the disease state as we had proposed in our hypothesis. But this is converting a healthy neuron to make it fire like a Fragile X neuron, which is interesting, but that's not what our goal should be. Our goal should be the converse, which is to see if we can reverse the aberrant firing pattern. So that's the next set of experiments Shreya did. She started recording from a cell, generated a baseline of firing patterns, then added the low 100 micromolar concentration of S100 beta onto the same cell, continued to record and see if and how that recording changes, the firing pattern changes. So the same experiment, but now with a low concentration of 100 micromolar S100 beta. So this is now Fragile X control neuron with control astrocyte, healthy firing pattern. With the lower concentration of S100 beta, nothing changes. It's already in a healthy state. So none of these parameters change. But when you take a Fragile X neuron in the company of a Fragile X astrocyte, this is double whammy. This is both cells are diseased now, and that is reflected in the aberrant firing pattern shown here in red. If you now add 100 micromolar low concentration of S100 beta, it starts firing like a normal neuron. So we are able to correct the aberrant firing pattern with a lower concentration of S100 beta. Therapeutically very interesting. Now this is another case where you have a healthy neuron, but in the company of a diseased astrocyte. So it fires like a diseased neuron. If you now add S100 beta at the low concentration, it converts it to a normal firing pattern. And if it's already in a healthy state, there's nothing to convert, so that firing pattern doesn't change. So this is then very, very interesting. A low concentration of 100 micromolar of S100 beta makes Fragile X neurons fire like controls. So we are able to reverse the abnormal firing in Fragile X neurons simply with this low concentration of one particular molecule that is produced in astrocytes called S100 beta, and the levels matter. The higher level is a disease-inducing condition. The lower level is able to reverse the disease condition and bring it back to the normal level of firing. So here then is basically, remember, we started our journey with showing that the genotype of the astrocyte was able to determine how the neuron will fire. So the astrocyte determined how the neuron will fire. Then we substituted the astrocyte with its rasam alone, and the rasam itself could determine whether the neuron would fire abnormally or normally. And now we are saying we have a further refinement. We have found a surrogate for the rasam altogether and picked one particular molecule secreted by astrocytes, S100 beta, and now show that if you take any condition such that the neuron fires normally, If you now add high level of S100 beta, it starts firing abnormally. And here you have a disease firing condition, which is the red Fragile X disease firing condition. It's already at a disease state. Adding high S100 beta makes no difference. It continues to fire abnormally. And then again, if you now take a disease condition, such as this one, a Fragile X firing pattern, and now add lower levels of S100 beta, you can convert its firing, you rectify the aberration, and make it fire like a normal neuron. And in a normal firing pattern, adding the low, of course, makes no difference. So just by varying the level of this one molecule secreted by astrocytes, S100 beta, we can bidirectionally flip the firing, just as we did with astrocytes, and with there it's rasam. So we have now narrowed it down to one molecule that seems to be doing the job, which is quite remarkable. But in biology, there are many ways at getting at a solution, right? There are many paths to reaching your goal, because it's a complex system with many molecules that can modulate function. So we may be very happy to find this S100 beta is correcting the defective firing, but is it doing it for the right reasons? Is it doing so by acting on the target molecule that we originally identified as being the problem child? Remember, I had told you earlier, remember this part, that there's an abnormal reduction in a particular type of current in Fragile X neurons, meaning persistent sodium current, INap for short. And I told you that in Fragile X neurons, this persistent sodium current was significantly reduced. So S100 beta may be correcting the firing pattern, but is it acting through that particular current? Is it exerting its correction by reversing that reduction in persistent sodium current? If it's acting in that route, then the prediction is that because S100 beta is fixing the problem, it should be able to reverse the abnormal reduction in the persistent sodium current seen in Fragile X neurons. So remember, I had shown you this complex chase earlier by applying this ramp voltage on neurons to specifically isolate this one subtype of ionic current called the persistent sodium current. And I had shown you this slide earlier. In a healthy neuron, here's what the current looks like. You measure this peak here, and in healthy neurons, and that peak is essentially missing. There's a massive reduction in the persistent sodium current in the Fragile X neurons. And we had quantified that in this graph by showing there's a reduction. Now, when we apply the low concentration of S100 beta, the same 100 micromolar low concentration that was able to reverse the abnormal firing pattern in Fragile X neurons, is also able to reverse the reduction in the persistent sodium current. So it brings back the persistent sodium current to normal levels, back up to normal levels. So it is indeed acting on the target that we had originally guessed and verified, and it by reversing the aberrant reduction in the persistent sodium current. So I'm going to stop here with some key conclusions for you, take-home messages for you. First of all, this is for the first time that anybody has shown that these human Fragile X cortical neurons show this dramatically aberrant burst firing activity, spiking activity that is quite distinct from healthy cortical neurons. You don't have to be a neuroscientist to see the difference between these two firing patterns. And quite remarkably, it is the genotype of the astrocyte, meaning whether or not the astrocyte is a healthy one or a Fragile X one, will determine how the neuron fires, whether it fires normally or aberrantly. So the genotype of the neuron can get overridden by the genotype of the astrocyte. So the astrocyte is boss, and it determines how the neuron will fire, whether it will be abnormal or healthy. Furthermore, we went one step further to show that you don't even need the astrocytes in the co-culture. You can just take its rasam, and that alone is able to show you the same switch from Fragile X to healthy and from healthy to Fragile X. Okay, so that's quite remarkable. That then led us to the search for something in that rasam that might do the job as well. And there we found that S100 beta that is at low levels secreted by the healthy astrocytes can actually reverse the abnormal firing pattern seen in the Fragile X neurons. And we also have a mechanism through which it does so, which is that it acts on the persistent sodium ion channel and reverses the reduction of that current seen in Fragile X neurons. So we have a candidate and we have its mechanism as well of how that reversal and correction can be achieved. So broadly speaking, taking a step back, almost much of the majority of the earlier research in the field of Fragile X has actually focused on synaptic changes in Fragile X neurons. That's the last 20 years of work has focused exclusively almost on how synapses, the connection between neurons, is altered as a result of Fragile X. More recently, people have started looking at excitability changes as well. But the focus has been on the neuron and what happens in the neuron, in its synapse or in its firing properties. What we have done today, what I have shown you today, is that you can go beyond that, well beyond that, and look at networks of neurons grown as a mesh in a dish, and then look at bursting activity across the network and find absolutely new kinds of aberrations that were not looked at in detail earlier. Some of it was done in mouse models and in the past, but nothing has ever been done in the human model in this domain. And most importantly, we are now on the verge of a paradigm shift because we are saying that all that goes wrong inside the neuron is not caused by things inside the neuron. It can be regulated by an outside agent, a next door neighbor, another kind of brain cell called astrocytes, which were not at all in the picture earlier. And so aberrations and their correction in the neurons are not cell autonomous, meaning they're cell non-autonomous effects, and astrocytes can exert their influence bidirectionally in this system. So astrocytes suddenly become a new player in finding new ways of regulating what happens in the neuron. And specifically, we identify a molecule called S100 secreted by astrocytes that goes and acts on the persistent sodium channel in the neuron to give you this switch, which means that therapeutically for the longest time, we have all been searching for a cure and ways of alleviating the effects of the disorder by looking in great detail inside the neuron. What I'm telling you today is that you can go to astrocytes right next door, and the astrocytes may hold a solution too. So the astrocytes can give you benefits that have not been explored before. So now the focus shifts to the astrocytes and see how those could be leveraged to find new treatment that can help reverse the defects in the neurons. So with that, I'll end by acknowledging the phenomenal work done by our team in NCBS Bangalore, Shreya Dasharma, who did all the electrophysiology, Raki Pal and Bharat Reddy, who did all of the hard work in terms of generating these different paradigms, these two protocols, and these platforms involving human neurons and human astrocytes, which is sort of at the core of the story. We were also aided greatly by our collaboration with the University of Edinburgh, specifically Professor David Wiley, who helped all throughout this work in advising us on the electrophysiology. Siddharth and Chandran, who is a stem cell expert and does disease models of neurodegeneration. His lab helped us with many of the protocols, and his colleagues, just such as Bhuvanesh, were very, very helpful. The original some of the cell lines we got from Emory and Cedars-Sinai. All of this work was enabled by generous funding from the Department of Biotechnology, originally from the DBT, and more recently from the Science Foundation. So I'll stop here. Thank you all for your kind attention, and I'm happy to take questions.
Thank you, Professor Shona, for the wonderful talk and a very clear explanation of what's going on. We have already received a few questions, so I'll start with the questions.
Sure.
So Arush has a question. Why are mouse astrocytes used?
Yeah, so originally our logic was reversed because we are still thinking in the conventional manner. So we thought we want to see what happens in the neurons and the Fragile X neurons. So we will have a pure system where we have healthy or Fragile X neurons, and let's keep the astrocytes the same. Just the mouse astrocytes are just a backdrop and a sort of a secondary cast, which will be there as a constant, right? And so we wanted to focus on the neuron. And we found these firing pattern differences, and we understood how they go wrong. All of that was fine. But then we thought, well, exactly the question that was asked, why mouse, right? Let's try the humans and see what happens. So when we switched to the human system, human neuron, human astrocyte, our first nervousness was, will we see the same effects? Maybe everything will just get thrown out of the window. So we were very relieved and assured when we saw the exact defect that we saw with the mouse one, that the same firing pattern differences emerged. But then we stumbled onto something quite dramatic, that the genotype of the human astrocyte was crucial, that we missed completely with the mouse astrocyte system. Yeah. So this is the trajectory of science, interesting twists and turns.
Thank you, Professor Shona. Next question is by Shamita Bishwas, and she asks, how are the action potentials measured?
Oh, they're measured exactly the way I showed you. Let me see if I can find. I actually don't have a slide, unfortunately. Let me see. Maybe I have a poor man's. Yeah. So if you look at this on the right-hand side, the green cells are the neurons and the orange cells are the astrocyte. This is the mouse astrocyte, by the way. So you have these very sharp glass pipettes that we fabricate, and the tip of the pipette is, 2, 3, 4 microns, tiny. And we fill it up with solutions, electrolytic solutions. And so then we guide this electrode onto the cell body. The cell body is roughly 10, 20 microns, okay? So under microscopic guidance, we can go in and target a particular neuron and touch the tip onto that neuron and break into it. So now you're in continuous contact with the inside of the cell, and you can measure whatever voltage changes happen inside the cell, and also you can control the inside of the cell. So this is the essence of what is called whole cell patch clamping, a technique for recording from individual neurons using electrophysiological recording techniques. These are tiny signals, as I told you. The currents are as tiny as picoamperes sometimes. So you have to have these fancy amplifiers that amplify the signal without amplifying the noise. And then you can get these signals out. And the signals can be either changes in voltage in the units of millivolts or changes in current, in this case, in units of picoamperes. So single cell recordings, yeah.
Okay. Thank you, Professor Shona. Next question is by Ananya Sachdev. You have already touched upon this in the talk, but I'll still ask this question. If it's the persistent sodium current, what's causing the short durations of the peaks? The repeated part makes a bit of sense, but I don't understand the less duration.
That's right. That's a very, very good question. Yes, absolutely, you're right. So we think, and we have evidence for this already, that there are other players involved to keep this talk simple. So there are BK channels, for example. We have already shown that they play a role. So the duration can be modulated with other ion channels. And so it's persistent sodium currents, BK currents as well. And we believe, to be very honest, there might be other players involved as well. And it's not exactly clear how S1 and beta go. It does not act directly on the persistent sodium current. It actually is, S1 and beta is a good molecule for buffering calcium. So it actually acts on calcium in the medium. And that change in calcium goes and acts on calcium channels, which in turn then causes these changes in bursting patterns. So there are more ion channels involved. And one of our first papers that we published with this, Shreya has actually gone into that and shown how those other channels change. And this is a paper in molecular autism in 2020. And you'll find those details there. But for this public lecture, I didn't want to crowd it with too many details of this kind. But it's a very good question. Thank you.
Thank you, Professor. Next question is by Aritri Malik. What if the unhealthy astrocyte rasam is there in presence of low levels of S100 beta? S100, yeah.
We already did. I thought I had shown that. Let me see. Did I miss that? It works.
Yeah, I think
I think I did it, but I have very poor memory, short-term memory. So is this the one here? No, not this one. Oh, this. Yeah. So you're right. I haven't shown this, but this is the one that would work, right? This is a control neuron with a Fragile X astrocyte. So if you now replace the astrocyte with astrocytic condition medium, you get the same phenotype. And if you now put this in, one would expect a similar effect. There are certain problems, and I think you're hinting at that, and let's try to expand on that. When we grow them together, they're in a separate dish, right? The medium or the astrocyte, whatever. And then we transfer one of these little cover slips with those neurons into the recording chamber, where they no longer are bathed in that original medium. They're now sitting in a recording medium, which is meant for recording, electrophysiological recording. It doesn't have any of the stuff that they had while growing up. And now we are adding in that blank slate, we are adding different levels of S1 and beta, right? So that's the distinction to be made. So we are never recording from a situation where you're swamped with the medium, the original medium, and then adding something. We let the medium do its thing in the neuron, then pull the neuron out, put it in the recording dish, and add blockers or whatever else we do in an acute experiment for a short term. So your question is well taken. If you were to go into the medium and start flooding it with different levels, what would happen? We haven't done that experiment. And also there's an associated question that you may have in your mind, which is how long do you have to do this for and things like that? How persistent is the effect? Those are experiments that we have to do now to get more clearer answers.
Oh, thank you, Professor. I thought you had shown this case, but anyways, I'll move on to the next question. The next question is by Anushka Nayak from JP Institute of Information. She says, I would like to thank you for such a wonderful talk. And my question is it possible to detect FXS before birth by techniques such as amniocentesis, and can external factors such as alcohol, certain diseases can cause FXS in fetus, or it is completely hereditary in nature?
Yeah, so there are prenatal screening techniques now that are available, at least in the US. And so then there's also genetic counseling that is done in terms of cases of parents where they may be at high risk. So that's all possible. As far as we know, this is purely a monogenic cause of autism. It's that specific mutation in that one gene that causes this aberration. So we do not know of no environmental factors are known to be causing this. I mean, the symptoms may get exacerbated and get worsened or something because of other factors, but the root cause is that mutation in the FMR1 gene, nothing else.
Thank you. I think she joined late maybe, but still I'll ask the second part of her question. As of now, do we have any techniques to extract live human brain cells for such experiments without causing any damage to the human body?
The answer in short is no. There are ethical reasons as well. But there are cases where it is done in extreme cases of disease where it may be fatal. For example, in epilepsy patients who are suffering from severe epilepsy, and none of the drugs work, one has to remove the offending tissue, the area of the focus of the epilepsy, surgically. If that area does not affect, surgeons will decide whether removing that part of the tissue would impair motor function or speech, and if the bar is pretty high. So if it's not affecting absolutely essential function, and it is crucial for the survival of the patient, people do remove epilepsy tissue from the brain. And this surgery is done in Bengaluru, for example. Some of my surgeon friends do this, and in that case, you get tissue out. And we have actually helped in doing some recordings from these brains, but the tissue in Bengaluru. But those are already diseased tissue. You cannot, and this is adult tissue, we cannot use those to generate stem cells. And in some cases of cancer, of course, glioblastomas, one has to do biopsies to get the nature of the exact kind of cancer that's happened, the staging and all that. In that case, people take a tiny part of the tissue to get, but that's again not for research, not very helpful.
Thank you. I'll move on to the next question. Next question is by Medha R. Can a diseased neuron be cured inside the interaction of neurons astrocytes instead of in vitro astrocytic rasam? I'm not sure if you understood the question. I think she's indicating that without the rasam, can you fix the this problem by
Yeah, I mean,
addressing the neuron?
the first data set that I showed was the actual astrocytes themselves, right? Just being in the same neighborhood, neurons and astrocytes together in the culture was enough to reverse it, right? What led us to the rasam is that what is doing that? It's great that the astrocytes do it, but how is the astrocyte doing it? That question led us to first saying, do you need the astrocytes physically to be present? Will the rasam be enough? The rasam was enough. What's in the rasam? And so on. But of course, in the real situation, you have the neuron and astrocytes together, growing together. And so that's there, of course. It is the search for what mechanisms can mediate this correction that led us to the rasam and beyond.
Thank you. Now I will take a few questions from YouTube live chat. The first question is by Akash. What are the effects of the aberrant spiking activity?
Very good question. Yeah. So you would notice, let's just take one of the older ones. Yeah. So you will notice that in normally, actually, this bursting pattern that you see, the normal bursting pattern shown here, a prolonged burst, but few of them with long gaps, is what actually you see even in vivo in animals, right? That's the normal pattern. So the fact that we are getting this in a dish is quite remarkable. And if you have these bursts, but separated in time, that means that they are they can go and activate a barrage of synaptic activity and then quiet. It's not hyperexcitability. Whereas if you keep firing lots of bursts without any sort of cessation of activity, that's hyperexcitability, and that causes aberrations in the brain. As I said, Fragile X patients often have epilepsy, and epilepsy is aberrantly high firing. So this may be an indication of that sort of hyperexcitability, which is the hallmark of autism. And if you constantly have this kind of barrage of activity without stopping, it will really cause excitotoxic damage in the brain tissue. And that's one of the reasons why this is bad.
Thank you. Thank you, Professor Shona. So there are similar questions, but there's a direct question by Nazneen about this. What is the role of astrocytes in the human brain in mental disease progression? Do they play a role and do they get affected by cosmic events, or does astrocytes have a clinical component? I think she wants to know a lot about astrocytes.
Yeah. I mean, I used to think of, and this is again a pun I can make only in India. Astrocytes have for the longest time been around. They've been known to exist. People studied them, but nobody paid any attention to them. They were like the OBCs of the brain. OBCs meaning other brain cells, right? It now turns out astrocytes are absolutely vital for the functioning both in health and disease of neurons. They wrap around synapses, so synaptic transmission is regulated by them. All kinds of ionic fluxes are regulated by them. The same receptors that are found at synapses and neurons can be found in astrocytes. So astrocytes do not play a supporting role. They play an important regulating role of how neurons function. This is now being recognized, and now it's an intense area of study to see how astrocytes go wrong. Of course, disease affects astrocyte. Even in the context of what I presented to you today, people have shown that astrocytes are affected in Down syndrome, for example, and astrocytes can play either a detrimental effect or a beneficial effect, depending on the case, situation of the disease model. So the fact that astrocytes can influence neuronal function in terms of a non-cell autonomous function is now being recognized quite widely. In the context of Fragile X and this kind of disease, this is the first such study, but there's a growing awareness that one has to not only understand what goes on in astrocytes, but the neuron-astrocyte interaction. There's another class of other brain cells, as I call them, which are called oligodendrocytes, which are again very, very important and now being sort of paid attention. Then there are microglia, which are also important. So it's a fuller picture now that we're beginning to get. And what was the other stuff
Yeah, the other stuff, are these astrocytes affected by cosmic events?
Not that I know of. I don't know what we mean by cosmic events, but maybe we can. I mean, the reason they're called astrocytes, let me clarify, because they look like stars. That's all. They have these branches which are like stars, and so anatomists name them astrocytes. Cyte meaning cell. So that these are cells that look like stars, and hence the name astrocytes. There's nothing astrological or astrophysical about it.
Okay, thank you. Thank you for the explanation, Professor Shona. So next is a related question. Avni asked, how did it occur to you that it is the astrocytes that should be used in the rasam experiment along with neurons, and not any other component of the brain, say any other kind of glial cells?
Yeah, it's purely in terms of where we wanted to start. We started only with neurons, then neurons with mouse astrocytes. Then for sake of completion, we thought we should do human neurons with human astrocytes. And the moment we did that, we found this astounding thing that the astrocytes are making a difference. And so then we went along that path to see, can astrocytes, are they playing a role, and exactly how are they mediating the role? So it was really a starting point for us, and there's many more possibilities. I'm sure there are other types of glia that could do similar things or more exciting things. But you should also be aware of the fact that growing and these healthy, robust platforms involving human patient-derived astrocytes and neurons is non-trivial. These are extremely finicky and sensitive protocols, extremely expensive as well. So the astrocytes take about 6 months to grow, and the neurons take about 3 months to grow. And if anything goes slightly wrong in the protocols in terms of contamination, the whole thing goes. And you have to feed them all those sorts of reagents. And they're very, very time-consuming and resource-consuming. So one has to start somewhere and take baby steps and get somewhere. And what I've shown you today is all these steps that we have taken. But we got lucky, we found something pretty cool. But I take your question in the right spirit, that this opens up an actual framework for looking at many different combinations. In fact, we don't know what's wrong in the astrocytes, right? It could well be that the neurons in turn could influence the astrocytes, and we don't know that yet. That's another line of investigation we'd like to pursue. But with the caveat that technologically this is extremely challenging, and it's a very, very sensitive platform that requires sort of a very high, it's a high maintenance system, let's put it that way.
Thank you, Professor Shona. There are a few last questions. So there are a few questions about S100 beta. So Akash Modaki asked, what is the function of S100 beta? And Joy asked, which receptor does S100 beta bind to?
Yeah, so S100 beta is a molecule that's been known to play multiple roles inside astrocytes, okay? In fact, S100 beta is used as a biomarker for astrocytes. If you want to see, if you have a bunch of cells and you want to know are they astrocytes, you could stain for S100 beta and confirm that they're astrocytes. So it's a very ubiquitous molecule specific to astrocytes, and it has multiple functions. What I have told you today is just one aspect of its influence on ion channels in neurons, very likely mediated through its actions on buffering calcium in the medium, the external medium, which in turn affects ion channels in the neurons. But it has many different roles, both in disease and in health, which I, of course, haven't covered, and there's considerable literature on it.
Thank you. Thank you, Professor. So there are a few questions related. I think you have already answered one, a similar question. Can FXS can be detected by any means like amniocentesis or CVS? Actually, even I don't know what does this mean. So if you could tell us.
Yeah, I think
how is it...
the prenatal testing that's done for Fragile X for unborn babies, I forget how they collect the samples. I'm just missing it right now. But there are ways of prenatal screening for Fragile X, yes. But the best way to do it, I think the most recommended and widely used way is soon after birth. If you start seeing signs of autism, such as by 1.5 to 2 years, you'll see certain external symptoms, such as the gaze aversion, not making eye contact with the mother, for example, avoiding eye contact, clasping and things like that, not clasping things, and other ADHD-like symptoms. Those will tell parents that there's something that's off developmentally in terms of developmental delays as well. And that's the point where most people go and do a screen to see what it could be. Fragile X is, of course, the leading genetic cause, monogenic cause of autism, but there are other forms as well. So it could be any one of those. The symptoms converge quite often between these disorders. But for Fragile X, if there's a good reason, genetic reason, because of the parents' genetic background, if there's any reason to be worried about potential Fragile X, in which case a prenatal screening is done in the West, much more widely available. Unfortunately, it's not that widely available in India, as far as I know.
Thank you. Thank you, Professor Shona. I think we have addressed all the questions which have been submitted on the chat, except, I had a question which I think is a little bit, but still I'll ask the question. So what would happen in a person or a child once this is fixed? I mean, if someone is having an aberrant neuronal activity and you somehow fix it, then does the brain resume back to the normal state? Or if that particular intervention is missed in a particular development stage, then it won't have any effect? I mean, it's again, I think it's a...
Yeah, that's a good question. So these are developmental disorders, right? Neurodevelopmental disorders. So one assumes that there's a developmental window where one can intervene to reverse course or at least change the trajectory. Now, right now, we don't have anything like that for any forms of autism. But cognitive behavioral therapy, CBT, which is often used, does help because the brain is very plastic in these young individuals. So if you intervene with certain behavioral therapies, it does make a difference, and it can change the course of the development of the disorder in many cases. There are windows, even beyond the developmental windows, one can continuously work on these individuals and help them acquire normal function, depending on the severity of the disorder. So I must emphasize that unlike Alzheimer's or Parkinson's and other neurodegenerative disorders, autism often is, the cells are not dying in these cases, okay? It quite often, in Fragile X actually, for example, there's an abundance of synaptic connections. There's too many synaptic connections. You just saw there are too many action potentials are being fired. So sometimes these disorders are disorders of abundance, meaning that something has gone above the normal range and you have to dial it down, which means that the ways are there to dial it down. We have to find those ways of dialing it down, right? So that gives me hope that in case of autism, and especially in Fragile X, we must find ways of dialing it down because it's not an irreversible damage that's happening early in life. It's just managing and putting it back on the rails to something in a normal range, right? So that's why I think fundamentally autism and neurodevelopmental disorders give us reason for hope. We just have to find combinations of drugs maybe that will help us do that. But cognitive behavioral therapy in conjunction with drugs is, I think, the way to the future.
I just got a message from Ananya saying that I have not asked her question. So I'll just ask this question. Did you investigate further what other molecules might act as S100 beta for playing a similar role in firing?
Very good question. We haven't, but we have some pretty good guesses. IL-6, for example, is one that we were close to testing, and then we found S100 beta to work. So there's a pretty good list of things that are good candidates. So with this framework that we have now set up, this platform, one can start running these other molecules, which is something that's quite doable, given that this platform is working rather well. But as of now, we haven't thoroughly tested these other possibilities yet. We have tried drugs that act on the persistent sodium channel and shown that those drugs, as predicted, do reverse the defects. So that gives you also a handle on the therapeutic aspect in terms of drug design.
Thank you. Thanks a lot, Professor Shona, for the wonderful talk and clear explanation and fantastic findings from your lab. Professor Spenta is also around if he has any question or comments. Professor Spenta?
Well, it's a great talk, Shona.
Thank you.
I had a question, but I'm very shy to ask it.
No, please don't. Please go ahead. Don't be shy. Come on.
A question like this that, you showed two types of firing rates of a cell. And there is a correlation to behavior. It's an amazingly mysterious thing that if the cell fires in a certain way, the child behaves in a certain way. And if it does not, then, avoids the mother's gaze, etc., etc. I mean, it's almost all of neuroscience is like that, actually. In some sense, things are firing, but maybe you cannot tell what I'm thinking about by just even knowing everything about the electrical activity of my brain. I think it's a very fascinating question, but maybe it's also a non-question. So I
think what you're hinting at is the fact that in the neuronal encoding of information, in terms of the long history you guys have hosted Bill Bialik in ICTS, right, in workshops, and Bill was one of the pioneers of this in terms of actual encoding of information, and there's information theory that comes in there as well in a big dose. But the fascinating part now, as you rightly point out, is that a lot of this was correlation earlier. Biologists always mistake correlation for causality, right? We are very good at that. But now we have a way of causally linking this, neuronal function to an animal behavioral function output with optogenetics, right? So you can actually specifically activate subtypes of receptors in specific circuits and modulate action potential firing patterns in these subsets of complicated circuits and modulate behavior. Or you can silence or activate and change addiction, change memory. You can create false memories in the hippocampus. It's stunning stuff, actually, right? So this is a very, very exciting time to do neuroscience. And if I may, since this is a much more mixed audience, is that, if there are young folks out there, physics is great, but time to move on to neuroscience, because we need those skills and those quantitative analytical skills that physicists acquire so naturally in their training. And I think it's a good time to have that brain power, no pun intended, focus on brain science, because that's where some of the most challenging and fascinating questions are waiting to be answered. And we now have the tools to do that with human cells, with animal cells, you name it. And I think this is a fantastic time for all the fantastic bright students that come through the Indian system, especially most of them go to physics or engineering. It's time for some of them to take a look at this. It's a good time to do that.
Yes.
So thank you for raising that.
So after all, biology is engineering in some deep sense, right?
It is, yes. Yes, absolutely, yes.
It's very different from
physics. I mean, all of this, as I showed you with this small example today, it's a reductionist approach. You start with one thing, you break it down, right? And that's something that we physicists and engineers do all the time, quite naturally.
Thank you so much. Thank you.
Thank you very much for hosting this talk. Thank you very much.
Thank you, Professor Shona. Thank you, Professor Spenta. Vijay is here and he has a remark. Please, Vijay, go on.
How was the birthday?
You're muted.
Vijay, you're muted.
Great. Sorry. Yeah, I'm back. Sorry. I just wanted to remark about the physics and neuroscience. So incidentally, we have a workshop on quantitative systems biology coming up in ICTS in December. It's a long 10-year-old school that's been running between ICTP and ICTS. And this time the focus is on neuroscience.
I know. One of my students has applied.
Yeah, on sensory motor control.
Yeah. So
I'm very happy to see that, yes.
I want to add one thing, that one of the speakers, Larry Abbott, actually was a high energy physicist.
Absolutely. And he has this book with Peter Dian called Computational Theoretical Neuroscience, which is fantastic.
Yeah.
So if you're getting Larry here, I don't know whether he'll be able to come or we'll do it online, probably online. Larry is fantastic. He's a shining example. He's a pioneer in the field. Yes.
Thanks.
Thank you. Thank you. Professor Shona, I had one last question.
Yes.
I couldn't, prevent myself asking this. So, as I understand this reverse engineering of stem cells to make them as human brain cells, and there's this term called organoids. Are they the same, you have been working with, or what is it?
What we have is a two-dimensional culture, just a mono layer of neurons and astrocytes in a dish. Organoids are the next step. You're now trying to create 3D tissue, little clumps of tissue, which have a z-axis. What I showed was only X and Y. If you now start adding depth to that tissue, you start getting closer to little pieces of brain tissue. And that's where now the new action is. But again, what I'm struck, I'm completely an outsider in the stem cell field. 5 years ago, I would have laughed if somebody told me I was going to do this work. But, you know, it's really Wild West. A lot of it's tinkering, trying things out. It's my uncle's recipe versus my aunt's recipe, and something works with you, something doesn't work with me. And so it's quite Wild West and quite uncertain. But through this sort of muddling along with these trial and error methods, new technologies coming up where organoids are being grown for disease models. And that's where the next frontier is. We needed single cell resolution to go after this. So for us, organoids will be the next step, but we'll lose some of the single cell resolution in these clumps of tissue. That's why it's a trade-off. If having 3D tissue is beneficial for answering the question, we'll go there. If not, we'll stay with this. So it's a decision each researcher has to make, depending on the question.
Thank you. Thank you, Professor Shona, for the wonderful talk. Thanks.
Thank you. Thank you all very much for joining on a Sunday afternoon. Take care. Thanks a lot. Bye-bye.
Thank you. Thank you, everyone, for joining.